Hello.
All right. So let's see. It's 9:39. I suppose we should wait for a minute. Are they going to tell us when to start, or are we live?
It said we were in a practice session when we joined, but then it's also recorded. So we are live now, according to Lee. Thanks, Lee.
Okay, thank you. Well, so let me start. It's 1 minute still left, but I think we had a very nice foundational lecture just now. All right, it's 9:40, so here I go. Let me begin by extending a very warm welcome to all of you to the first keynote lecture of ISBA 2020, being held right now virtually. It is wonderful to have so many of you attend this event after what has been and continues to be immensely challenging times. In terms of logistics for this address, I would encourage all of you to keep your microphones muted when the speaker is speaking. And please feel free to post your questions or comments in the Q&A box, which I will try to entertain at the end of the lecture. The lecture will last for about 50 minutes, and we expect about 10 minutes for questions and discussion. Our speaker today, Amy Herring, really needs no introduction to ISBA. She's someone I've known for over 20 years as a truly wonderful friend and colleague. In fact, Amy and I graduated in the same year, yet I'm simply blown away by the quality and productivity of her research. She is without doubt one of the most influential biostatisticians of our generation, with over 275 scholarly publications and an H-index bordering around 70. Amy currently holds the title of the Sara and Charles Ayres Professor of Statistical Science and Global Health at Duke University, and has made a profound impact in a number of areas, including, but by no means limited to, complex dependent data analysis, longitudinal data analysis, and Bayesian dimension reduction methods. Amy's contribution to Bayesian modeling have found their way to numerous applications in health and medicine, and particularly so in the environmental health sciences. Going through the numerous academic honors and awards that have been bestowed upon Amy will take up the entire hour. So let me just note the Mortimer Spiegelman Award from the American Public Health Association, perhaps the flagship award in biostatistics, her fellowships of the American Statistical Association, and more recently, the Janet Norwood Award for outstanding achievement by a woman in the statistical sciences. But perhaps even more importantly, Amy is the epitome of a perfect citizen for our profession, and her invaluable generosity in serving ISBA is almost legendary. Ladies and gentlemen, please join me in welcoming a truly exemplary scholar and citizen of ISBA, Amy Herring, as she presents Bayesian modeling to predict chemical toxicity using high-throughput screening data. Amy, it is over to you.
Oh, thank you so much, Sudipto, for that really wonderful and kind introduction. I think everybody has people in the field they look up to, whether they're their seniors, and in the case of a lot of the JSBA leaders right now, they're juniors. And Sudipto has been somebody that I feel like is one of my batch mates, even though I didn't have the pleasure of being at ISI. I hope to visit one day, but I really just try to be more like Sudipto every year. He's just a wonderful person, and I appreciate and I'm honored to be in the room with him today. I'd also really like to thank all the volunteers and the organizers of this meeting. The organizers essentially organized 2 or maybe more conferences, first with boots on the ground and then going virtual, and it's a lot of hard work. I really appreciate that, and I also appreciate the help I got from the volunteers in learning how to navigate this session. Hopefully I learned well, and I won't embarrass my teachers as we go along. So with that, I will go ahead and share my screen. I'm supposed to share my entire desktop, so let me just do that and see. Sudipto, is that good? Okay, wonderful. Thank you so very much. So I also, while I'm in the mood to thank people, would like to thank my collaborators on this project. They span a number of disciplines and all played a really important role. First and foremost, I'd like to thank two graduate students, one actually now a scientist at Los Alamos National Lab who did all the heavy lifting. In the top right, you see Kelly Moran, that's her outside the door to her thesis defense. She did the work that I'll be talking about during the first part of the talk. And then in the middle, you see Bora Jen, who's a current PhD student who did some of the work I'll talk about a little later. And these are just two incredibly brilliant and hardworking women I've had the pleasure and joy of working with here at Duke. And so they get all the credit. They'll both be speaking later during the conference, Kelly in the Savage Award application session, and Bora also on different work. So I encourage you to check out those sessions. They'll both be talking about methods for Gaussian process regression. Then we also worked, because the data are very complicated, we worked with a couple of experts in the area. Stephanie Engel is an epidemiologist. She's on the far left. And in the bottom right, you see two toxicologists, David Reef at North Carolina State and Julia Rager at UNC. Finally, we have two other statistician collaborators or partners in crime. And the bottom left is Matt Wheeler, who's at the National Institute of Environmental Health Sciences. And the top left is David Dunson here at Duke. So I'd like to thank all of them for all their hard work on the project. So what I'd like to talk about today is spend a little time explaining toxicity testing paradigms. And one of the reasons I'll spend a lot of time on that is that these data are freely available through an actually fairly easy to use R package. And so this is an area that has a lot of interesting problems. I feel like we're only scratching the surface. And so I'd love for some other researchers to get involved and help us by solving some of the other problems in the data. So I'll talk a good bit about that. Then we'll talk about ways to exploit chemical structure to predict responses. And then we'll talk about, in some of the work Bora is now doing, how we can use data on multivariate responses available in some of these studies. And at the bottom here, you see the ToxCast robot. So these are some of the high throughput screening chips on the left and robots on the right that are used to carry out the experiments we'll be talking about for the most part today. And then just for a little background, I want to explain for people who may not be fully aware of how regulation happens for chemicals in the US. And the short version is it's very complicated. The Environmental Protection Agency does a lot of the regulation, but some of it goes through a variety of different agencies like the Food and Drug Administration, for example. So it can be a little confusing, but back in 1976, US Congress passed the Toxic Substances Control Act. And that act was a little odd in the sense that it regulated introduction of new chemicals and regulated existing chemicals. But when they passed the act, they grandfathered in all the chemicals in existence as safe. So that's what they did. They have gone back now to test some of those chemicals, and 5 of them have been banned subsequently. So one thing to note about the US is that we don't like to ban things here. There'll be a plan to prioritize chemicals. So there are over 80,000 chemicals currently in the inventory. The EPA puts them in some sort of priority order. They will do one set of experiments. And at that point, they will either conclude the chemical's low risk and stop testing. They'll conclude they don't really know enough and put it back in the queue for further testing, or they'll identify a risk. Even when the risk is identified, it doesn't mean the chemical will be banned. So in general, they prefer to talk to companies and have companies on their own decide to pull a chemical off the market or use something different without putting in force a ban. But they also can take regulatory action to outright ban a chemical. So that's how the process works in the US compared to the rest of the world. So while I'll be talking about the testing paradigm in the US, we're certainly not alone in testing chemicals. And this plot just shows pesticide testing in some nations and areas with large agricultural presences. You can see that the United States has only banned 37 pesticides from use compared to 47 banned in China, 71 in Brazil, and almost 170 in the European Union. Interestingly enough, there's very little overlap across all 4 of those agricultural reasons. Part of that is due to differences in climate, crops, and organisms that affect the growth of the crops, but other factors, including a reticence in the US to ban things, are also at play. So how are chemicals tested? So if I had seen Shelley's talk a little earlier, I might have actually tried to find some video because car talk is hard to beat and follow up. But mainly these chemicals are tested are not in humans. So getting human data is very challenging. I'll talk about that a little later, generally because controlled exposure studies are viewed as in general not ethical, and so they're quite rare. But the three main sources of information are in vivo studies, and these are often done in animals, primarily rats, but the animal model may depend on what the outcome of interest is. In vitro studies, these could be done the old-fashioned way by humans in a lab, or they could also be done using more modern high-throughput settings, type experiment settings. The latter are harder to control and tailor, but you can do them a lot more cheaply and do a lot more tests at once. And then we'll be talking a little bit today about in silico methods, trying to use what we've learned from experiments and the power of computing to make predictions. I will point out in terms of animal studies, since we don't have a social event here at ISBA this year, there's a wonderful children's movie that came out a long time when I was a child called The Secret of NIM. And I didn't know until I was in grad school that NIM was the National Institute of Mental Health in the US, which funded at the time animal research. And it's a really scary children's movie, and I encourage you to check it out. It's very good. The animators were great. It's not a Disney thing. It's more of a nightmare-inducing thing. But it's focused on animal research and the ethics of animal research in a fairly large way. There's also a series of novels that are very good upon which the movies are based. So if you want a social event, that's a great one. So here's the problem with human data. So here's a plot showing about 30 chemicals. These were measured in pregnant women's urine who had decided to participate in the Mount Sinai Children's Cohort study of neurological and potential effects on child obesity due to exposures. And you can see with the dark red, these are large positive correlations in exposure. So if we look at free-living humans and we try to determine, what kind of chemical exposure might have caused a developmental delay or overweight status, it is very difficult to pinpoint one specific chemical. The correlations here among several groups of chemicals are quite high. So it's almost hopeless. I won't say entirely hopeless, but it's very difficult in human data to pick out one specific exposure that may have caused an outcome, or even one specific exposure that plays a major role, because the exposures tend to be quite highly correlated. And 30 is not a large sample size for these experiments. I'm now working with an investigator at Duke who's putting little wristband, like the little plastic bracelets on people, and they're measuring thousands of compounds on those bracelets. Sorry. So here's what these screening devices look like. So on the left, you see plates. And so these high throughput screening assays are fast and cheap. They were originally developed by biopharmaceutical industry to help in drug development. And you can see here a 96 well up to a 1500 well plate. And these plates will contain matter such as biospecimens or proteins, and they can be interrogated with a large number of chemicals and doses of those chemicals simultaneously. To give you a sense of the scale of this testing program, the Environmental Protection Agency's Toxicity Forecaster or ToxCast program has data on roughly 10,000 chemicals and over 1,000 different assay endpoints, so different proteins, different receptors, et cetera. Versus fewer than 600 studies being conducted in live animals by the National Toxicology Program. So they've gone very strongly in this direction of high throughput studies where they feel like it's feasible. So what kind of information is available in the data? So there's a lot, and I'll focus today on three big components. The first is the assay endpoint. So we will initially focus on the Adagene PXR assay. And so this assay uses human liver cancer cells. They're actually harvested from a 15-year-old boy in Argentina back in the 70s. And this cell line's been cultivated since that time, and there's a lot known about this cell line in particular. And they're looking at perturbation of transcription factor PXR, okay? And so what does that do? Basically, that transcription factor senses the presence of foreign toxins and upregulates expression of proteins that are involved in detoxifying and clearing things from your body. So the key takeaway point about this assay, and the reason it was chosen in a paper by Matt Wheeler back in 2019 and chosen by us, is that, as one might expect, given its function, it's reactive. So it's more easily turned on than some of the other assays. And so we expect to see lots of signal for the adagene PXR. We'll look at some other assays later in the talk. The other very important piece of information is the chemical itself. And the structure of the chemical is something we'll talk about exploiting in order to get insight about toxicity. One thing to keep in mind with ToxCast is that the selection of chemicals into the database is informative, which is good. We wouldn't want our resources being wasted by randomly selecting chemicals for evaluation. In the first phase of ToxCast, they picked pesticides with extensive prior animal data, and that was because they wanted to do proof of concept work, to be sure these high throughput methods were giving them answers that they expected. The second phase went on to some data poor chemicals of regulatory or research interest. For example, a lot of green chemicals were included in that second phase. And then in the most recent phase, they've been looking at flame retardants and endocrine disruptors, for which there was interesting epidemiologic data available that spurred them on to do more mechanistic type studies. And then the dosing is interesting here. The chemicals are tested at a variety of dose levels. They're typically half-log dilutions, but sometimes you see sparse or irregular spacing. They're about 50-something unique doses represented in the data set. There is some replication, but it's not as much as you might think in the data set. And so here's a visual of the data. We can see along the vertical axis, the chemicals, along the horizontal axis, the assay endpoints. The predominant orange colors are showing very few replicates for most of the combinations of chemicals and assay endpoints. So the data are quite sparse, and you can see a lot of white in the middle, representing no information about that combination of interest. The vertical black line at the left is the adagene PXR assay that we'll be talking about for most of the talk today. And you can see it's been pretty well interrogated across the different chemicals as well. So here are two chemicals and the dose response information available in the data. So on the right, you see the chemical structure, and I'll talk a little bit more about that as we talk about how it's quantified. On the left, we see the dose response. So we can see in the top left, chlorobenzolate has been fairly heavily interrogated. It does look like there is a dose response relationship there with higher levels of response associated with higher doses of the chemical. On the bottom left, you see a chemical that has been much less heavily interrogated. There are only 3 data points, and doesn't seem to be a whole lot going on. They're right, right sitting there around 0. So this is typical of the variability in the data. And then what do we mean when we talk about chemicals being similar or dissimilar with respect to structure? So on the left, we see 2 similar chemicals, 2,4-dimethylphenol and 3,4-dimethylphenol. And on the right, we see 2 chemicals that we would call dissimilar. So that's maybe simple to tell based on the picture. But what do we really do? So we'll take chemical BPA. So I'm sure in the US, at least, people are used to seeing plastics that say BPA free. It was a common chemical used in plasticizers and found in water bottles, among many other things. Here's the chemical structure of BPA. And there are many ways actually to take a chemical and to quantify it. And this is a really interesting area. We have faculty in the math department here at Duke working on methods to do this. And there are a variety of different canned methods, including Mold2, which is the one we use to extract quantitative information about a chemical. So this is something I would find really interesting to play around with some more and to see what we learn using other ways to quantify a chemical. Lots of times these chemical quantifications are based on a SMILES, which is a representation you can see below. And based on that SMILES, it's possible to extract 777 chemical features. And so here are some examples, counting the number of oxygens, the number of aromatic rings, the molecular weight, and so forth. One thing to note about these features is that the pairwise correlation can be quite high. So here's an illustration of that. And many of these descriptors are driven by molecule size. So the count of carbons, the count of oxygens, the molecular weight, the number of atoms, all of those variables have pairwise correlations above 0.9 in the data set. So we do have these descriptors, but a lot of them are describing underlying concepts such as size. One thing to keep in mind as we think about this 777 dimensional space is that the direction of maximum variability in the molecular features isn't necessarily the direction of most relevance to the toxic response. So there could be things that chemicals have in common that are completely unrelated to their toxicity for the endpoint we're interested in. And so that's what this graph shows. Each point is giving the Euclidean distance between the functional principal component scores in the response and PC scores in the structure that accounts for 95% of the variability in the data for one pair of chemicals in structure and toxicity. So we don't see a really remarkable straight line kind of relationship here. So something to keep in mind. So what are our goals? We'd like to learn a low dimensional space that underlies both chemical structure and activity. We'd like to develop a distance metric for chemicals, relying on the structural features relevant to toxicity. Okay, so what's a toxicity relevant distance, and can we use this to try to inform future testing strategies? We need to be able to handle sparse data, irregularly observed dose response curves with some replications potentially. And then we'd like also to be able to make predictions for new chemicals not yet represented in the data. So let's talk about some of the work that's been done. And there is a broad class of models that have been developed called QSAR, quantitative structure activity relationship models. And for example, Locam et al. In 2015 developed a Bayesian regression tree approach. In this case, each leaf on the tree represented a different dose response surface. This method was used to learn the relationship between chemical properties and dose response. However, the issue here in our case is that it lacks the ability to scale to the number of chemicals and molecular features that we're interested in. Matt Wheeler has a nice paper that appeared in 2019, I think in Biometrica, where he developed a Bayesian additive adaptive basis tensor product model. It was based on Gaussian processes. In terms of our work today, it relies on a pre-processing step to compress the predictor space using naive PCA. So it could be possible that that naive PCA doesn't capture, in particular toxicity relevant factors. And in addition, it has some scalability issues. However, I will note that Kelly Moran is presenting on Thursday, and she's presenting joint work with Matt Wheeler that's talking about ways to get around scalability issues in Gaussian processes. So I guess stay tuned for that method. It may be looking a whole lot better if we can use some of their newer work. And then other really interesting work along these lines has been done by Wesley Tansey and co-authors, and they recently updated their paper on arXiv. They've done really nice work in the context of candidate drugs for cancer treatments. And so they're considering, in addition to things like the drug, the dose, the tumor type, site information, such as information on genomic features, and they discuss in future applications, they might be able to take into account information about the chemicals or even the mechanisms by which the drugs work. So that's something definitely worth keeping an eye on as well in this area. Finally, other methods, one we'll talk about relating our work to later today is sparse function on scalar regression models. They can select important chemical structure features and give them appropriate weights as we describe the dose response. But in this case, because we have a lot of potentially highly correlated features that may have some redundancy in them, a principal components analysis-esque type approach might be more appealing or more informative than some sort of penalized regression strategy. So here now, I'll finally get to the model and tell you a little bit more about what we're doing. And we call this Bayesian partially supervised sparse and smooth factor analysis, or BS3FA is the name that Kelly gave to the method. And so again, our goal is to relate chemical features to the dose response. And so if you look on the right side of the figure to the red lines, each line is a dose response curve. And each row there in the matrix corresponds to the dose response curve for a different chemical, okay? And so if we look in the top right of the figure at that first red line, we can see a relatively flat relationships to start. And then we see about halfway through the dose scale, some kicking in and evidence of a potential toxic effect. Then in the second figure, we see an example where we have a pretty quick increase in the response over the dose range, and then potentially some sort of, certainly a change in slope and maybe a flattening off. And then in the third row, we don't see very much going on, okay, and so forth. And so these are our responses. And so if we look to the left of them, well, then we can see a matrix that represents our chemical features. And so here we'll stay the 777 chemicals that we've measured, or features of the chemicals that we've extracted using mold 2 to quantify the chemicals. And then for each chemical, we'd like to be able to link the dose and the response to determine if the chemical is affecting the adagene PXR assay. So if we look at the very top, you can see we have these latent terms eta, and these lines are now representing probabilistic dependency. So the latent eta terms impact both the chemical structure and the dose response components of the model. So if you really want to see the mathematical expression, that's down now on the left. And what this model assumes is that all structured variation in the dose response curves are explained by the term lambda eta I, where lambda is a typical loadings matrix and factor analysis, and the epsilon is just random noise. And below that equation, note that the chemical features X, and now you can see I've introduced notation Z. That's because these features are not all continuous or symmetric. So I might have counts of atoms of a certain type. And so we need to introduce underlying latent variable Z to facilitate our modeling of mixed scaled chemical features, okay? So for example, if Z is positive, then that might lead to a value of 1 for a binary feature. So that's all that notation does. So now I've switched to Z, and we can see that eta is not only in our model for the dose response, but also in our model for chemical features. And so it's a shared latent variable between the two models, but the loadings matrices are different. Also note the new I term in the features model allows us to address systematic variability in the chemical features that's unrelated to the toxic response. And so because of the structure of this model, our latent variables have nice interpretation. So eta has an interpretation as a latent variable giving us information about the toxicity relevant variability in the data. And new is giving us information about the toxicity irrelevant variation in our feature space. And this is an advantage over simpler methods such as naive PCA, which may assume all the variability in the feature space for the chemicals is potentially related to the toxic response. All right, so let's get into more details about how we fit the model. And first, we'll think about lambda and the prior for lambda. Now, because Y is a functional response as a dose response curve, we want the columns of our factor loadings matrix lambda to be functional in nature. And so because of this, we're going to model the columns of lambda as coming from a Gaussian process. The term lambda eta ends up being the sum of multiple functional curves, and therefore it's another function. We're going to use a multiplicative gamma process shrinkage prior developed by Anirban Bhattacharya and David Dunson, so that as we get further and further over in the columns of the lambda matrix, we're getting closer and closer to a flat line, and essentially we're just trying to avoid pre-specification of the number of factors K in advance. So instead, we're going to specify an upper bound on K, and then we're going to use this prior to handle the extraneous factors in the model. Now, what do we do for theta? So we take a similar approach specifying the prior for theta, but because our chemical features aren't functions, we don't need a Gaussian process here. However, we do want sparsity in theta because there may be a number of factors that are unrelated to a particular chemical's structure. And so to encourage this behavior, we put a horseshoe prior on elements of theta. And again, we're going to apply shrinkage on successive columns using a multiplicative gamma process prior. Now, after accounting for variability in our chemical structure space that's shared with Y, We also now want to address structured variation in the chemical feature space that's unrelated to toxicity. And so here we're going to directly apply the Bhattacharya and Dunson prior for the elements of the matrix C. So here's just a little more information about the shrinkage, and note the column-specific shrinkage variance component, tau K inverse, is shared across the loadings matrices, okay, for lambda and theta, which is allowing us to learn the dimension of this latent space simultaneously. All right, so how does this work in practice? So we did two broad classes of simulation studies, kind of the typical, if you think about as a grad student, for those of you who are grad students listening, how do you decide about a simulation study? Well, you simulate under your model, and then you simulate again under something else. And so in this set here, we've simulated data from a partially shared latent factor model, and we also did simulations that are available on arXiv and on the online material for our paper, which is currently in press, under a model that's not a factor model. In all cases, we held out a quarter of the chemicals to assess predictive performance. So we held out entire chemicals, not just certain doses for chemicals, to try to be more true to life to the situation where you have a brand new chemical and we don't know what it does. Okay, so what do we have here? We are exploring performance measured by distance in the toxicity relevant space in the true data versus the estimated model, where we have the toxicity relevant factor number, K, increasing as you move to the right across the separate squares, and the toxicity irrelevant factor space, J, varying within each square, okay? And we're going to compare our method, which is denoted here, BS3FA, to function on scalar regression, to the principal component scores that define 95% of the variation in the data, and to Euclidean distance in all the factors, okay? And you can see at the top, the red line, that's us, that's Kelly's model. It does quite well across all values of K and J, so this is good. We can see PCA struggles. It struggles more as the number of toxicity irrelevant factors increases. Again, this is the behavior we're expecting to see because it's treating all that as in the same space. And that deterioration as J increases also occurs for the other methods. And even to some extent for our method, though it is a little bit more robust. Now, not all methods allow for us to assess prediction for new chemicals, but here we're going to compare mean square predictive error for our method to Bayesian and frequentist function on scalars regression, and to Matt Wheeler's method, which here is denoted BAA-BGP, and the lasso for each chemical dose, and two-way interaction between chemical and dose for the lasso. So you can see here that we're doing pretty well. If looking across the bottom, the mean square predictive error does deteriorate for all the methods a little bit as the number of irrelevant factors increases, but Bayesian function on scalars regression and our methods are tending to do a little bit better. We can also look at coverage of the predictive interval and the holdout data. And here we see some evidence that Bayesian, that function on scalars regression is quite conservative. So its coverage is close to 1. We're approximately on target. And so the basic moral of this story is that we're feeling pretty okay moving ahead to analyzing the data from ToxCast. So what do we make of the data analysis? So first, let me tell you exactly what we did. We pulled out 3,100 chemicals for analysis. We withheld 25% of them, again, to see how we do if we see, quote, a new chemical. But we did include the features of those chemicals so that we could make predictions. We just held out the dose response for that 25%. And the learn matrix lambda gives us some information on the structured variation in the dose response data. So here we can see, if we look at the figure, the top middle figure labeled first column of lambda, this is a very prototypical dose response curve, okay? Starts out nothing going on relative to control. So 0 would be relative to a control dose in the experiment. That's that dotted red line. And then we see things go up, and then we see a little downturn at the end. And that's interesting and something you have to be a little bit careful about with these data. Sometimes what you can have happen in these experiments, and even though we've used methods suggested by the EPA to screen for it, we can still get a little bit of it. It's called a cytotoxicity, and that's when the dose is so high that it essentially blows up the cell. It destroys the tissue, and you don't get a very meaningful measurement of the response you're interested in. This is particularly a problem with these high throughput technologies because you don't have really specialized control over the dosing. And so once you've run the experiment, you have to go through with the advice of a toxicologist and determine which experimental responses are valid to use and which seem to show that the entire tissue sample was destroyed, and then that dose and higher doses should be ignored. So that's something to keep in mind. Later columns are going to just provide smooth deviations from our overall shape. And so what we can do is we can look at, now I'm going to focus on that first feature, the kind of prototypical dose response curve, and look at the values of eta. And that will give us information about how each curve is weighted in the estimation of a specific chemical's dose response curve. So who's got a big value of eta 1? I can look at those chemicals and then look at the theta matrix to look at the properties of those chemicals that have big loadings there. And you can see them listed at the bottom of the slide. All of these chemicals are known toxins, and you can see some of their characteristics that had large loadings in theta, things like the number of chlorine, and then basically a lot of other chemical features. And I had chemistry when I was 16 years old, so I'm not going to try to explain to you exactly what those mean, but they are associated with the toxic chemicals at the bottom. So we were feeling like the method is certainly being reasonable here. Now we're looking at prediction of activity. First, one thing to think about is if we look at the chemical, the eta distance here. So before we think about predicting for these chemicals, if we were thinking about designing a new study, how might we pick new chemicals to test? And so one thing we could do is look at chemicals that we know are toxic and fill in the space around them. So who's close to those chemicals in toxicity relevant space that we don't know anything about? And the idea would be then that we're trying to figure out who the other bad guys are, okay, who are related to the criminals we've already identified. But we can also take a completely different approach and say, well, here if we look at the eta distance, there seem to be a number of chemicals that aren't close to anything. And so in that case, we know very little about their potential toxicity. And so we might want to move and interrogate that space as well, looking at quite dissimilar chemicals from well-characterized compounds. And in reality, we probably want to do a little bit of both, right? If we know something's bad, we want to know if small deviations from it are also bad. And if we don't know anything about a chemical, it seems important to learn more. So here are some chemicals that we considered activating that were in our held out sample. And our definition of activating was that the bands of the 95% predictive interval shown here in dark gray for the predicted dose response curve mean excludes 0 at any point. Okay, so the dotted red line, horizontal line is no effect. The blue represent the observed data points. And you can see for the most part, we do fairly well. For chemicals that we don't do well, they tended to take on shapes that were not characteristic of other chemicals in the data set. So if you look at bisphenol B, we see a really sharp increase in the dose response. And so we called it active, but we underestimated the activity compared to what we see in the data. And then here we see some chemicals that we would call non-activating, and that's because the predictive interval across the mean never excluded the value 0. And you can look at the data points, and for the most part, it looks fairly null, or we don't have a lot of data to help us make this decision. And so now what I'd like to do with the time I have remaining, and oops, I just unplugged myself, which is really bad for my laptop because it's been crumpy, is go back to this full feature space in ToxCast. And you can see here We've just been talking about for quite a long time, only one assay endpoint and one little line there on the left. And there are tons of different assay endpoints. So how can we use this information to learn more about potential toxins? And this is where Bora's work has come in. And we're really interested in trying to make connections between these laboratory-based toxicity studies where you have a cell or a protein exposed to only one thing at a time. And we'd like to be able to relate that to the human study I showed you before, where we saw that matrix and these women were exposed to tons of correlated chemicals. So how can we move towards using that to learn about what's going on in humans and to try to figure out if there's an adverse response and we have high levels of exposure to 10 correlated chemicals, which one might it be? Can we get that information out of the data somehow? We're not going to go there today, but that's somewhere we're definitely interested in going and would love to collaborate and talk to people about that. So as in Bora's work, we took a little bit of a step back. We're going to use more endpoints than the Adagene PXR, and we're also going to take a step back in how we define activity. And so in the work we did with Kelly, we used the standard definition of activity that's used by the EPA, which is a change in the mean dose response, essentially. And based on our collaboration with toxicologists, really any disruption in the dose response is considered a change in activity. And so that could also be a disruption by increasing variance with increasing dose. So that type of heteroscedasticity is really common in studies of this type. And so we want to be sensitive to those types of departures from the status quo. We like to be able to predict dose response and activity for non-tested pairs and eventually to prioritize chemical for further study. And so motivated by an epidemiologic study, the Mount Sinai Children's Cohort, which looked at outcomes in children, including neurodevelopmental delays and childhood weight status and obesity, we're going to pull 131 assay endpoints that are relevant to neurodevelopmental disorders and 271 assay endpoints of relevance to obesity. And when I say we're going to pull, that wasn't like a half a day task. That involved a lot of meetings between Bora and toxicologists and toxicology grad students to try to look at molecules that serve as biomarkers in those pathways that are related to disease etiology or that are therapeutic targets for treatment. And the chemicals of interest were chemicals that were of interest to the epidemiologists that they had the ability to measure in their study. So they included pesticides, phthalates, which are part of actually the new car smell, going back to car talk, and some other chemicals of interest. Of all the possible chemical by endpoint combinations, just over 40% had no data at all. So a big missing data problem there. And here's a snapshot. So if we look across columns on the right, these are the assay endpoints. And this is after talking to toxicologists about how to make them a little bit more understandable. So, sorry, I guess, is the moral of the story there. The chemicals you can tell a good bit about. So we have bisphenol A here. We have DDE, which comes from DDT. That's a chemical we all know. And so you can see that for some of these chemicals, we have a lot of interrogation on some endpoints. And then in some places, we have a lot of missing data. And even a chemical we know is bad, so DDE is no good guy, it doesn't necessarily activate all these endpoints. So we see some activation, but not activation across all of them. So we'd like to be able to fill in the missing squares to get some sense of what's going on there. And so to give you a sense of what our model is, now we have the response, which is a fold change relative to control. We have a response for chemical I, for dose, for the dose XIJK, K could be, or K represents potentially repeated measures, and J represents the assay endpoint, okay? And then we have a binary variable, gamma ij parameter that takes value 1 if the average dose response is non-constant and 0 otherwise, okay? So this is just telling us whether there's disruption in the mean response. And we'll get to the error structure and the reason for it in just a bit. So what does the EPA do now when they decide whether a chemical is active or not? So here's a figure of 3 models the EPA fits to these data. So the first across the bottom, that's not the x-axis exactly, I guess. That is what they're calling the constant model. Okay, so no change in the response from the control condition. Then they'll fit the popular Hill model, which you can see the equation given in blue. This is a nonlinear model that allows for sigmoidal shapes. And then also the gain-loss model, which is an extension of the Hill model that gives more flexibility in the form of the dose response. One reason these are popular models with the EPA is that they have direct parameters in the equations here that have nice interpretations in terms of toxicity relevant values. But one challenge with these models, which you might be able to tell based on their fractional forms, is that fitting them can be really unstable even when the data aren't sparse. So it can be a bit of a flaky business. One nice thing about our approach is that we are Bayesian, and if we want to get at some functional of interest to a toxicologist, for example, the AC50, which is where the response hits 50% of the maximum response seen across the experiment, we have samples from MCMC and we can easily get to this. So what EPA does is they fit these three models and they have a variable in the data set called a hit call. That's sort of their analog of the switch gamma IJ that I defined on the previous slide. And it will be called active if either the Hill model or the gain-loss model has a better value of the Aki-Aki information criterion to that of the constant model. Okay, so this is just a simple AIC-based measure. Not calibrated, just calculated. So when we think about how we want to set up our model, here are some of the more interesting dose response shapes we see in the data. The primary goal, again, is to detect any disruption of the function by dose. We're not that interested in interpreting the function directly, okay? So we have to remember, if we're thinking about, what's dangerous to humans, that we're not going to get all that excited about this dibutyl phthalate, increase, downturn, and increase again, because we don't even know how relevant that assay endpoint is to humans. We might not expect the exact same dose response shape. We're just looking for any disruption, okay? So any sign that something's been changed. Because it is kind of difficult to think of an overarching model, and certainly based on these shapes, we wouldn't want to assume monotonicity. We're going to use an unrestricted B-spline basis with coefficients assigned to prior that allows us to borrow information within assay endpoints in this model.
Amy, just a quick note, probably you have another about 6 to 7 minutes to wrap up.
Perfect.
Just letting you know. Thank you.
Thank you so much, Sudipto. So here we can see some evidence of heteroskedasticity. So here are two chemicals that have been really strongly interrogated in the data set. They don't seem to do a whole lot, at least diisobutyl phthalate doesn't do a whole lot in terms of the mean, but we do see clear evidence that the variance is increasing with the higher doses. For the chemical on the left, it looks like we do maybe see a downturn as well as some variance increase. And so we're going to define an analog to our gamma switch T that takes value 1 if dose is related to the variance and value 0 otherwise. And so this broadens the typical definition of activity that's considered in the standard software and used by toxicologists in order to determine whether a chemical is active or not. And so we're just going to adapt our initial model. What we're going to do now is take our variance component, sigma ijk squared with an asterisk on it, and put a log linear model on it to allow that variance component to depend directly on dose, okay? So, so this is really all we've done, and in a lot of ways, it's a simple and clever idea that Bora had. I don't have time to give you all the details of the simulation study, but I can tell you that when we borrow information across chemicals and across assay endpoints, we get lower mean square error and much greater in-sample area under the curve than the standard models that the EPA is using, even in the gamma parameter, even in just prediction of activity in the mean, because we are borrowing information across chemicals and endpoints there. And so here are some results. And what I'm showing you here are the fitted curves as well as EPA's hit call. So whether EPA calls it active, and then the probability, posterior probability of disruption in the mean and the variance estimated from our model. And these are two known bad guys, okay, bisphenol A and DDE, which is a breakdown product of DDT, which actually has been banned. And you can see here for bisphenol A, a clear increase in response with dose. Our model identifies that there's a very high probability of disruption in the mean and very low probability of disruption in the variance, which is consistent with a figure. If you look on the right, we're estimating a relatively flat dose response. EPA did call this chemical active, and so they do find evidence that there's disruption in the mean. What we do see is low evidence for a disruption in the mean, but high evidence for disruption in the variance. And so it may be that the EPA's hit call has been influenced a little bit by the heteroskedasticity here. Now I'm going to look at two examples, bisphenol A again, but now with a different assay endpoint and also one of the phthalates, where our determination differs from that of the EPA. So in both cases, EPA called these chemicals inactive. In the first case, we're calling a mean effect for dibutyl phthalate, and on the right, we're saying there appears to be both an effect in the variance and in the mean. And if we look to see, well, who's right? Are we right? Is the EPA right? Well, that's a little hard to tell with the data we have without being able to run more experiments. But if we look at the in vivo animal studies, we can see indication that dibutyl phthalate and bisphenol A do have relevant toxic effects in the pathways we're interested in. So it seems like our response, our model's output is more in line with different data sources from the whole animal experiments that have been conducted. And so to wrap things up, I think there are a lot of really interesting related methods that are important in numerous contexts. So, and as I mentioned earlier, I really like this work that Wesley Tansey has been doing. That's really interesting. There's even somebody in the session I'm chairing later this afternoon who has a method that seems like it would be really related, and I'm interested in hearing that session. If you're interested in ToxCast and in toxicity, the TCPL-R package is what you can use to get at the data. And there are other ways to get at the data, but this is a fairly straightforward one. But I would just caution you that you do need to read the user's manual, and you may want to consult with a toxicologist so that, for example, you're able to exclude those doses above the level of cytotoxicity where the dose is basically blown up the cell. And then future work that we're still thinking about / banging our head on the wall about is how we ever use animal data to help us figure out what's going on in humans without making our priors overly informative, but also getting valuable information from them to try to sort out issues we have in the chemical mixtures setting where we have high dimensional exposures and really complex interrelationships of the exposures with behavioral factors and social factors as well. So the whole situation becomes much, much more complicated when we move to the human setting. And that's something we are interested in. So if you'd like to learn more, Kelly Moran's paper is in Press at Annals of Applied Statistics. I think you can see it online early and also on archive. Bora Jen's paper is still undergoing review, but she has an update recently posted on archive with accompanying our packages. And so thank you very much for your attention this morning, afternoon, evening, and dead of night. It's really nice to be part of this almost 24-hour-a-day conference. And I welcome your questions.
Thank you very much, Amy, for a wonderful talk and a very interesting talk indeed. We do have a couple of questions here, so I'm going to start. The first one is from an anonymous attendee. You have an option of disclosing who you are, I believe, but that's okay. In the interest of time, I'll read it out. What are the interpretations when the dose response curves are not monotone? That is, sometimes they increase and then decrease. Also, what is the indicator of toxicity in the dose response curves? Thanks.
Okay, so that's a great question. And actually, this is a fun part of the toxicological literature because there's sort of a wacky subgroup, and this is being recorded and I just called it wacky, but of people that believe in a phenomenon which is still debated called hormesis, which is a little bit of this is a good thing and a lot of it is a bad thing. And so an early downturn is something that would be consistent with that hypothesis that a little bit of some, smoke a few cigarettes and maybe it's good, and smoke a lot and maybe it's bad. That's the kind of thought behind that. And then when we get to the upper levels, then what we would typically, the most common thing we would see is this cytotoxic response, where the dose is so high that now I'm not measuring this transcription factor anymore because I've killed it, and so I have degraded the quality of the protein or the tissue too much to get an accurate measure. So, so for that reason, and for many reasons, because this is also like, this is the liver cancer cell of a 15-year-old boy who, was sick with cancer in the 70s, or these are Henrietta Lacks's cells, or some of these cells come from other, they come from a variety of tissue types. So like brain, liver, breast, they come from a variety of organisms. There's, mostly rats, but there's hamsters, there's pigs, there's even, I think, a boar represented in ToxCath, in addition to some human cells. We don't get all that excited about the shape. We just look at whether it's different from a flat line. So is there anything going on with this tissue that wouldn't be going on when it's exposed to the control substance?
Thank you very much, Amy. There's another question from Terence Savitsky. Terence, it's wonderful to have you attend the talk, and hello. And Terence's question is subgrouping of DR dose response curves relevant for dimension reduction?
So here we're not, again, we're maybe not as interested in sort of what cluster a dose response curve falls into. I think a clustering response is of potential interest. I think especially if we're looking not at these animal features, but we have human data, where I'm looking at maybe in the Mount Sinai children's cohort, I have growth curves. And so there I would be really interested in common features of the curves, because it might be that certain children, depending on their exposures, are not experiencing the slimming down that a lot of kids get when they're like 3 years old, and hitting, for example, growth curves in puberty at different times. And so I think that's so kind of taking a clustering approach, I think, is really interesting when we start thinking about analyzing the human data in particular. And thanks, Terence, for one of the people I miss seeing at this meeting.
Yes, thank you, Amy. I think these are the two questions I have from the attendees. I probably will take an opportunity to ask you one quick question. I was fascinated by the research. I was just wondering your use of the spatial factor models. So factor models are definitely very useful for dimension reduction, as well as for understanding or capturing the variability among the chemicals. And you have shown quite convincingly that how much better you can do in terms of principal components analysis relative to PCA. But I was wondering if the main inferential aim is about understanding or estimating the associations between the chemicals in this large collection. Would it perhaps make better sense to try to model them using conditional dependencies and sparse precision matrices, perhaps? Do you think that could perhaps even provide us with an opportunity to use some prior information about the relationships that may exist between these chemicals? Thank you.
So this is what you get for giving a talk on Gaussian processes of one of the world's foremost experts on Gaussian processes. So,
no, I'm just learning. I'm learning. Yeah.
So yeah, I'll have to think about, one thing that I think is really interesting is how distance has been measured here. And I think there's some much more interesting ways to break down the distance between chemicals, such as some that Jinping who in the math department here at Duke has been working on. So I definitely think that's one of the areas where there's a lot of space, to move in this project. But I have to think in a lot more detail before I give your question the answer it deserves, Sudipto, but I appreciate it. Somebody who's doing something interesting, I don't know. Laura's talk might be interesting for you for somewhat related reasons later in the conference, because she's been thinking of sort of interesting features of Gaussian processes that maybe aren't directly relevant here, but
no, sure. I mean, it was an honest question. I also don't know I don't have much of an intuition about this, but I was just curious in terms of the application and the modeling. But it was a fantastic talk, Amy. And I think in the interest of time, we that's pretty much all the questions we had, and it was absolutely fascinating. So thank you so much for your time, and thank you to all of you for attending this wonderful keynote lecture. There are many more exciting talks today as well as in the coming days, and I think so far I'm glad to see that the app is working well, and so let's hope it goes that way. And wherever you are in the world, thank you for attending, and please do stay safe and healthy. Thank you so much.
Thank you all. I look forward to your talks.
That was wonderful.
Thank you.
All right. Yeah, go ahead. You were saying.
I was just saying, I think we'll sign off so that this mail becomes available for the next session.
Right.
All right. See you.
See you. Bye.